The W and T genes have been characterized in vertebrates and invertebrates but seem to be inexistent in unicellular eukaryotic plants and prokaryotic. The W and T gene encodes for secreted proteins involved in intracellular signaling. These proteins play crucial roles in cell differentiation during development. W and Ts are 39 to 46 kilodalton proteins with cysteine-rich domains with 23 and 24 residues and 350 to 400 amino acids with N-terminal peptide. W and T signaling is a conserved pathway involved in cell differentiation and fate determination during embryogenesis and late stages of development. W and T signaling constitute one of the most critical biological processes during cell fate assignment. W and Ts are secreted glycoproteins that bind to transmembrane receptors called fissile. W and T proteins also interact with co-receptors of the low-density lipoprotein-related proteins. These co-receptors allow the Wnt pathway mediators to bind to other receptors. W and T signals are transduced through at least three distinct intercellular signaling pathways, including the canonical W and T beta catenin pathway, the W and T calcium pathway, and the W and T planar cell polarity pathway. W and T transduction needs binding to one of two family receptors, the fissile family and the lipoprotein-related protein family. Receptor fissile was identified in Drosophila, and then several homologs were identified in vertebrates. Fissile receptors consist of 7 types belonging to the G protein-coupled receptor family. The extracellular N-terminal side of the fissile contains a rich cysteine domain able to directly interact with the W and T protein. The intracellular C-terminal domain of the fissile contains a conserved sequence that interacts with another signal mediator named disheveled or DSH. The lipoprotein-related protein is considered as a fissile co-receptor. It is a transmembrane protein with an extracellular epidermal growth factor-like domain involved in Wnt protein binding and signaling transduction. The cytoplasmic C-terminal domain contains 5 conserved sequences required for W and T signal transduction. In the cytoplasm, several proteins are involved in the W and T cascade. DSH, a downstream of fissile receptors, is recruited by the C-terminal side of the receptor, leading to its phosphorylation. Another protein called axins interacts with C-terminal domain of co-receptor lipoprotein-related proteins and antagonize W and T signaling at the intercellular level. Beta-catenin is a key mediator of the W and T canonical cascade. Other than being involved in the W and T signaling, beta-catenin is in association with cadherin, plays a critical role in cell-cell interactions and cellular adhesion. It acts in association with beta-catenin and it binds to actin network and adherence. The beta-catenin levels responsible for either W and T signaling or cellular adhesion are constantly balanced. Soluble W and T proteins activate the pathway by binding to receptor fissile. This interaction can transmit signals via at least 3 distinct cascades according to the involvement of the beta-catenin protein. As mentioned before, they can be classified as the canonical W and T beta-catenin signal or the non-canonical W and T calcium and W and T PCP signaling. The non-canonical cascades are activated independently of nuclear beta-catenin, leading to different phenotypical responses. W and T soluble proteins are the initiators or pathway but have distinct effect on their target. Some of these proteins are responsible for the activation of the WNT beta catenin signaling, whereas others are responsible for the WNT calcium PCP activation. In Figure 2, we can observe the WNT mechanism when the WNT binds to the fusyl receptor activating the lipoprotein-related proteins, creating a beta-catenin cascade, which is translocated to the nucleus and interact with the transcription cell factors. In this presentation, we will be talking about the mesenchymal TGF-Beta signaling, orchestrating dental epithelial stem cell homeostasis through the Wnt signaling. A mouse incisor grows throughout the animal's life because of a pseudostructure at the proximal end that houses two types of tissue stem cells, dental epithelial stem cells and dental papilla mesenchymal cells. The dental epithelial stem cells are proposed to reside in the outer dental epithelium and the stellate reticulum and give rise to the inner dental epithelial cells, which are considered to represent transit amplifying cells. Stellate reticular, outer dental epithelium, and the inner dental epithelium together form the so-called cervical loop of the incisor. Inner dental epithelial cells then differentiate into ameloblasts and produce the enamel. The dental papilla mesenchymal cells reside within the papilla mesenchyme between the lingual and labial cervical loops, which can form the whole dental pulp mesenchyme, including the odontoblast. Previous studies have demonstrated that dental pulp mesenchyme controls the cell renewal of the dental epithelial stem cells and determines cervical loop size via mesenchymal epithelial cell interactions mediated through a gene regulatory network involving activin, follistatin, bone morphogenetic protein, and fibroblast growth factor signaling. WNT signaling is important for cell renewal and differentiation of various adult stem cells. In the canonical WNT beta catenin pathway, the interactions among WNT proteins, fusyl receptors, and low-density lipoprotein receptors, leading to a stabilization and translocation of the beta catenin to the nucleus, which then activates the transcription of target genes involved in the cell cycle reentry. In mouse incisors, the expression pattern of the Wnt-associated genes around the cervical loops imply the suppression of the Wnt beta-catenin activity during the maintenance of the dental epithelial stem cells. Continuous activation of the WNT beta-catenin pathway in the dental epithelium induces ectopic and continuous tooth formation in mice. Transforming growth factor beta signaling plays an important role in a broad range of cellular processes. Genetically modified mouse models have confirmed the cell autonomous requirement of TGF-beta signaling during the initiation step of both incisor and molar tooth germs. Here, transforming growth factor beta type 2 receptor TGF-br2 was specifically deleted within the postnatal dental papilla mesenchyme. This was used to understand the molecular mechanism underlying the cooperation between mesenchymal and epithelial cell.
I will be discussing the results, beginning with the TGF-br2 deficiency in mouse dental mesenchyme, which causes postnatally malformed incisors. They wanted to assess at first the mouse incisor expression pattern of an established CRE transgenic line, OC-CRE, which was previously used to delete SMAD4 genes in molecular odontoblasts. They used LAC-Z to stain mouse incisors of the OC-CRE and ROS-A26 double transgenic mice, which showed positive staining in mature odontoblasts of newborn postnatal day 0 mice, but not in the labial or lingual dental epithelial cells. What they found unexpectedly was at postnatal day 7, lax-Z staining had emerged within the interjacent papillomesenchyme between the lingual and labial cervical loops where potential DPMCs reside, and after extended throughout the dental pulp of the mesenchyme. In situ hybridization results also confirm that the CRE recombinase expression in papillomesenchymal cells, as well as odontoblasts in OC-CRE and ROS A26 incisors. What they also found was that TGF-Beta 1 was exclusively expressed in DPMCs, implying that it has a unique role in mediating signaling during the incisor development. We crossed the OC-CRE with TGF-BR2 conditional knockout mice to obtain OC-CRE TGF-BR2 mutant mice. In the Western blot analysis, it showed that this protein level was dramatically decreased in the incisors from mutant mice, and that the phosphorylation of SMAD2-3 was consequently decreased in mutant incisors. All of these special mutant mice displayed gradual abraded and shortened incisors after the age of 2 months, and had almost lost the outer portion of the incisors by 8 months, as you saw in the previous slide. In histological examinations of these incisors, the mice showed uniform constructions, but in contrast, the mutant incisors represented a strikingly thickened dentin layer on both the labial and lingual sides. In particular, there was a wavy mineralized structure that sprouted from the labial cervical loop region, and it was observed in the 3-month-old mutant mice, and more wavy mineralized structures emerged after that age. By staining the dental epithelial cells with keratin 14 antibody, it was found that this mineralized structure was composed of odontoblasts, ameloblasts, and their corresponding dentin and enamel layers. Higher magnification views of the labial cervical loop showed remarkably reduced ODE, stratified epitheliobloods at the outer portion of the cervical loop, and multiple layers of dental epithelia in mutant incisors. You can also see in this picture as well, they show it, that the histology of the incisors was restricted to odontoblasts but not dental papillomesenchyme, and found no obvious abnormality in mutant incisors in spite of the short stature and paralyzed limbs. So it suggested that the TGF-BR2 in DPMCs, rather than the odontoblasts, it contributed to developmental formities in the bone. So here you see on this slide figure 3 in our paper, and it shows the increased differentiation of DESCs in the cervical loop of the mutant incisors. Picture A is used to mark our LRCs. B through D are immunofluorescence staining of the BRDU and the Chi-67 in the cervical loop regions, illustrating that after 2 months of chase periods, BRDU is retained at the proximal end of the labial, OD in control incisors, like you see in B, whereas a few LRCs localized beyond the cervical loop in mutant littermates, reaching below the ameloblasts of the SI layer. In C, you can see that the BRDU positive cells in red are, and the Ki6 are green, which are negative, and at 4 months, LRC are hardly detectable at the labial cervical loop of the mutant incisor, which is D. And that's why it's boxed, and you can kind of see what's going on. The arrows are indicating the LRCs around the stratified epithelial bud. Panels below are higher magnification views of the framed regions in B through D, so it's just a better explanation of what's going on so you can see it a little easier. In E through P, it's explaining the in situ hybridization analysis of the expression of marker genes for DESCs, ID cells and ameloblasts, as well as odontoblasts/SI cells in the cervical loop regions of 2 to 4 month old mice. The black arrows in E through G point out the ODE, where the blue arrows indicate the ectopic DESCs in mutant incisors. The blue arrow in M indicates an ameloblast. We're going to do just a quick talk through of the increased differentiation in the DESCs in the cervical loop of mutant mice. They wanted to detect whether the abnormalities in mutant incisors were the result of aberrant functional activities of the DESCs within the labial cervical loop. What they found was that when it was applied to mark the potential DESCs located in the ODE from the control mice at age 2 and 4 months, the incisors from the 2-month-old mutant mice retained the BRDU label cells, which were localized not only from the ODE region, but also under the ameloblast and stratum intermedium SI layer. But by age 4 months, they had almost disappeared from the ODE of the mutant incisors and only sparsely distributed around the proliferative stratified epithelium rudiment. Then their other question in regards to that was whether abnormal differentiation was responsible for the decrease in DESCs in the OD region of the mutant mice incisors. They found that the differentiation of odontoblasts and SI cells were accelerated, as indicated by the expression of AKP, which is a marker for both odontoblasts and SI cells, which all of this suggests increased differentiation of DESCs, leading to the malformation of mutant incisors. Okay, so the next part is basically about the activation of the LRP5/6 of the beta-catenin signaling in the cervical loop epithelium of TGF-BR2 deficient incisors. They wanted to figure out if the Wnt/beta-catenin signaling is activated in the cervical loop epithelium of the mutant incisor, but compared to the control incisor, the mRNA levels of the LRP5 and LRP6, which encode co-receptors of canonical Wnt beta-catenin pathways, were dramatically increased in the cervical loop epithelium for mutant mice. Whereas when they found that the expression of RO2 and Reich, which encode receptors for non-canonical Wnt beta-catenin pathways, were not changed in the cervical loop epithelium for mutant mice. Therefore, the expression of axin 2 and the ubiquitous target of the canonical Wnt beta-catenin pathway was induced throughout the cervical loop epithelium of the mutant incisor. Furthermore, the nuclear accumulation of beta-catenin protein was observed in the epithelium cells of the stratified structure from mutant mice. So the findings strongly indicated that the mesenchymal TGF-Br2 deficiency led to the malformed incisors by overactivation of the LRP5/6 mediated Wnt beta-catenin signaling in the cervical loop epithelium. So the picture explains this activation of this signaling pathway in the cervical loop epithelium. And A through H is in situ hybridization analysis of expression of LRP5, LRP6, ROR2, and axin 2 in the cervical loop region of a 3-month-old incisors. The panels below them are higher magnification views of the labial cervical loop regions. So the arrows will indicate ectopically induced expressions of the LRP5, LRP6, and axin, respectively, in the cervical loop epithelium and or in the papilla mesenchyme. I and J are double immunofluorescence staining for beta-catenin and Chi67, which demonstrates that the beta-catenin expression was upregulated in the cervical loop epithelial cells and translocated to the nuclei of the stratified epithelial cells in the 3-month-old mutant incisors. The right panels are higher magnification views of the cystic rudiment frames in J. So if you take J and take it apart, you're seeing these pictures for the ones in the red box on the right. They really like to magnify everything so that you can kind of see what's going on, and that way you have a better understanding of what's being more expressed and what's being underexpressed, and so on and so forth. So the next part will be about the mesenchymal TGF-Beta deletion, activating dental epithelial LRP5-6 beta-catenin signaling by mesenchymal Wnt5A and FGF3/10. So they wanted to detect the expression of Wnt ligands, which contribute to elevated beta-catenin signaling in the cervical loop epithelium. Among the Wnt ligand genes expressed in the mouse incisors, Wnt5A was the only Wnt ligand gene that significantly deregulated upon TGF-Beta depletion. It was broadly expressed in the dental mesenchyme, except for the region adjacent to the cervical loop epithelium in the control incisor, while the mutant incisor expression was significantly elevated, especially in the papilla mesenchyme and follicle mesenchyme surrounding the entire cervical loop epithelium. They wanted to examine the increase of Wnt5 expression, which directly resulted in the impaired TGF-beta signaling, using AFI gel agrospeeds pre-soaked in an inhibitor of TGF-beta signaling, which is SB431542, and it was placed on the pulp mesenchyme of the ex vivo cultured incisor explants. Whole mount in situ hybridization demonstrated that this inhibitor induced strong Wnt5 expression in the pulp mesenchyme. It also, in cultured MDPC23 cells, the cell line derived from the dental papillae of phenomice, suppression of this TGF-beta signaling by this inhibitor upregulated Wnt5 expression, which indicated that disruption of this beta signaling promotes Wnt5 expression in the dental mesenchyme. And the best way to explain it is in this picture, which shows that it explains the mesenchymal TGF-Beta deletion activates dental epithelial LRP5-6 beta-catenin signaling by mesenchymal Wnt5 and FGF3/10. So in A and B, you see the in situ hybridization analysis for expression of Wnt5A in the 3-month-old incisors. The arrows indicate the enhanced expression of Wnt5A throughout the papilla mesenchyme and the follicle mesenchyme surrounding the cervical loop epithelium. And if you look at C and D, it's a whole mount in situ hybridization of P5 incisor explants, demonstrating induced Wnt5A expression within the pulp mesenchyme around the bead soaked with that inhibitor I was speaking about. So E is a real-time PCR of Wnt5 and MDPC23 cells treated with TGF-beta and or that inhibitor for 12 to 24 hours. Where n equals 6. F through I is in situ hybridization analysis for expression of FGF3/10 in 3-month-old incisors. Arrows indicate reduced expression of the FGF3/10 in pulp mesenchyme adjacent to the cervical loop, and J is the real-time PCR assay of the FGF3/10 in MDPC23 cells treated with the TGF-beta and or the inhibitor for another 12 to 24 hours. And K is the trowel-type tissue culture system for P5 incisor explants. The AFI gel beads are placed between the labial and the lingual cervical loops, so that's what you see. And then for the L through P is in situ hybridization analysis for expression of LRP5 and P5 incisor explants treated by beads soaked with growth factors or signaling inhibitor for 36 hours. The arrows will indicate induced expression of LRP5 in the labial or lingual cervical loops. Q is real-time PCR of axon 2 in P5 incisor explants treated with growth factors or signaling inhibitor for 12 to 36 hours, again where n equals 6. So the last part of our results are the haploinsufficiency of Wnt secretion, partially rescuing the malformed incisors caused by TGF-Beta deletion. The pictures are very descriptive, so I'm going to go through this really quick so that I can explain that to you. So they could clarify the involvement of excess Wnt signaling. They specifically block the Wnt secretion from pulp mesenchyme by concomitant depletion of the Wnt-less, an essential Wnt ligand transporter. First, Wnt-less was expressed throughout the dental epithelium and mesenchyme, except for where potential stem cells resided in the control incisor. Although the expression level of Wnt-less was not significantly changed in the mutant mice, the transgenic mice incisors, the number of Wnt-less negative cells in the ODE and papillomesenchyme were significantly reduced. Because of the shortened lifespan of these transgenic mice, they were analyzed at the age of 8 months, and they found that haploinsufficiency of Wnt-less partially rescued the phenotype observed in the mutant mice, which was evidence of the strikingly reduced mineralized structures. And the findings confirm that excessive Wnt signaling from dental mesenchyme contributed to the formation of wavy mineralized structures in these transgenic incisors. However, the contribution of elevated LRP5 and 6 expression to the phenotype of these transgenic mice still needs to be investigated, and that's what they found. So the picture is extremely descriptive. It shows the control as well as the different changes with the transgenic mice. So it's the haploinsufficiency of Wnt secretion partially rescues the malformed incisors caused by this TGF-Beta deletion. A and C are double immunofluorescence staining of the BRDU and Chi67 in the cervical loop region of the 3-month-old mouse incisors, which indicate LRCs in the yellow arrows within the OD and interjacent papilla mesenchyme between the labial and lingual cervical loops. B and D is in situ hybridization for Wnt-less in 3-month-old incisors. The red arrows indicate Wnt-less negative regions in the OD and interjacent papillomesenchyme. In E through N, it's the gross appearance of the outer portion of the incisors and histological analysis of incisors from the 8-month-olds of the different types of transgenic mice. The red arrows will indicate that the heterozygous reduction in Wnt-less significantly decreased the number of wavy mineralized structures caused by this deficiency, the TGF-Beta. The blue arrows denote the shrunken cervical loops in the OC, CRE, TGF-Beta transgenic mice, basically all the transgenic mice, and statistical analysis of the quantities of wavy mineralized structures in incisors from 4 different genotypes of 8 months. So in conclusion, we wanted to say that the findings showed that dental mesenchyme-specific TGF-Beta deletion results is upregulated in the expression of Wnt5A and downregulated in the expression of FGF310 in mesenchyme, which leads to the activation of LRP5, 6 beta-catenin signaling in the cervical loop, which also results in the postnatally malformed incisors by accelerating the differentiation of the DESCs. A previous study has demonstrated that ALK5 mediated TGF-beta signaling in the dental mesenchyme is required for embryonic formation of the incisor cervical loop. So the work attests to the important role of DPMC, specific TGF-Beta mediated TGF-Beta signaling in regulating the postnatal homeostasis of adjacent DESCs. Therefore, the deformity results from the increased differentiation of DESCs and is largely the result of impaired TGF-Beta signaling within the DPMCs. So to go with this, it's been realized that the TGF-Beta 1 was exclusively expressed in the DPMCs, which supports this unique role of the TGF-Beta signaling during incisor development. We also provide evidence that the LRP5/6 mediated Wnt beta catenin signaling promotes the differentiation of postnatal DESCs. TGF-Beta2 deficient incisors displayed overactivation of the LRP5/6 mediated Wnt beta catenin signaling, which specifically is in the cervical loop epithelium, and was supported by the upregulation of the LRP5/6 and axon 2, as well as the nuclear accumulation of beta catenin. What was really important was that it showed that partial ablation of the Wnt signaling by concurrent deletion of the mesenchymal Wnt suppressed the formation of wavy mineralized structures caused by the TGF-Beta2 deficiency. Mesenchymal TGF-Beta2 deficiency led to the increased Wnt5A expression and decreased the FGF310 expression, specifically in dental mesenchyme adjacent to cervical loop. This regulation of Wnt5 and the FGF310 expression of TGF-Beta signaling was seen in the ex vivo cultured incisor explants and the dental mesenchymal cell line. It was confirmed that the cooperation between the upregulation of Wnt5A and the downregulation of FGF signaling in the mesenchyme induced the activation of the LRP5/6 beta catenin signaling in the cervical loop epithelium. However, in the in vivo loss function experiment using the LRP5/6 knockout mice, it'll probably be more helpful to see that and to pinpoint the actual mechanism underlying the phenotype of the OC-CRE TGF-Beta2 mice, the transgenic mice. So they showed that the DPMC specific knockout of the TGF-Beta2 results in increased differentiation of DESCs, which then leads to the formation of wavy mineralized structures in the postnatal mutant mice. So based on those findings, they presented the relationship among various signaling pathways in the context of TGF-Beta2 deficiency and confirmed that TGF-Beta RLL-mediated TGF-Beta signaling in the dental papilla mesenchyme maintain the homeostasis of DESCs via regulation of cervical loop Wnt signaling.